Okay, last day, guys. Well, second last day, whatever. So let's get started. Shall I start? So we're going to talk a little bit about genome evolution and the origins of novelty today. And again, as is my past stuff, is mostly going to be empirical and data kind of a talk. What I want to give you is a flavor of a different aspect of using population genetics. What we've done so far is, can't hear me? Better? Okay. Right, so the way we've discussed population genetics so far, or the use of population genetics so far, is to understand or to be able to predict what are the dynamics that happen in future given some allele frequencies in populations, and the traits or what happened in the past and so on, right? But the traits we've always considered so far are phenotypic traits, right? Things that we care about, such as hair length, color, whatever, right? A behavior of an animal, phenotypes that are sort of discernible. I think one other aspect of population genetics, which merges very nicely into molecular evolution, is to understand whether genes and genomes themselves can be considered as traits that are under selection that can be evolving. Yeah? So hopefully some of this becomes a little bit clearer to you as I go along today. And so I want to touch upon, like I said, aspects of population genetics or applications of population genetic understanding, which blend with understanding molecular evolution. So I'm going to start off by discussing a little bit of work on synonymous versus non-synonymous mutations. So in Mridula's first talk, for those of you who are not quite familiar with this, you went over the fact that the genetic code is arranged in triplets, right? So there are three bases which form an entity, a unit, and those three bases are shown in this table as your first letter, second letter, and third letter. So UUU would encode a particular amino acid, phenylalanine. There are 20 such amino acids in most life forms. And so there are multiple such triplets, right? And so what's obvious to most of you already is that there's redundancy in the code, right? Which means because we have 64 possible codons, triplets that can be formed, there are 20 amino acids, many codons can encode the same amino acid, right? And so this is something that is largely conserved throughout life. So what we think is the genetic code was sort of frozen or set or had evolved in its major form very, very early on in the evolution of life. And later there have been some small tweaks and modifications as we went along, okay? So this table effectively summarizes what we call the generics or the standard genetic code, telling us which triplets encode which amino acid, okay? And so again, you see that some of them are two-box codons, which means that two codons, so one triplet is called a codon. Two codons both encode phenylalanine. Similarly, another two codons here encode leucine, but there are four other codons here which also encode leucine, okay? So leucine then is a six-box amino acid. It has six codons that will all give rise to leucine during the process of translation. So I'm very clear with this, especially the people who are not biologists. Yeah? Okay, and so now this becomes interesting because your naive expectation would be any organism, anytime it wants to encode, let's say, phenylalanine, should use UUU and UUC about equally, right? Randomly. Any one of them is fine because they both give you the same amino acid. Does that make sense? So mutations that will flip between UUU and UUC, so basically a mutation at that last position here, should be what are called synonymous mutations, which means the meaning does not change, right? Because they're both accounting for phenylalanine. However, if you now get a UUU to UUA mutation, you now have a change in the amino acid, right? So that would be a non-synonymous mutation. Everybody good with this? All right. So why do we care about this and why does this matter, right? The first set of observations early on when people started getting some genome, not even genome, gene sequences, right? In the 60s, 70s, 80s, as people started saying, okay, here's the sequence of, do you know what was the first sequenced organism? Virus, right? It's a bacteriophage that was sequenced first. And so its genome was small and people could sort of piece together the whole genome because it was tiny. It was incredible amounts of work just to get that one tiny genome. And now this is something that, it's done, right? I mean, not quite, but yeah. So, so as soon as people started having some of these data, it became obvious that naive expectation I just told you is not correct, right? Most organisms show a bias in their use of codons, and they seem to preferentially use some codons over others. And this has been a big mystery people have been sort of obsessed with for a long time, to understand why is there this bias? Why do we see this bias? Right? Because your expectation, as I said, is it doesn't matter. Everything should be about equally used. Any codon that encodes, multiple codons that encode different amino acids. So here's a sort of recent paper that shows what this codon bias looks like. So these are different genomes of Vibrio. It's a bacterium. And they're focusing on leucine codons. So leucine is a 6 box, yeah.
So there are some amino acids that have, like, as you said, 6 tRNA that codes for a particular amino acid. So this uneven distribution somehow reflects the composition of amino acids in the whole protein?
Overall, yes. That the amino acids that have more codons encoding for that amino acid tend to be the ones that are more highly used. Okay. In general.
So this would happen because of selection or some problem?
Yeah, there is some reasonable evidence that selection has shaped the evolution of genetic code, but it's not clear, for example, why UUU and UUC should be phenylalanine and not leucine. Okay. Right? And so that part, again, there's a lot of work on this. I just have no time to go over it, but it is absolutely fascinating, and it's really fun because you have no idea except for time machine to really address these questions, right? So you're sort of making your best guesses, your best inferences, given how we think selection works and how mutations are lost or not, and so on and so forth. So there is a lot of really fascinating work. I definitely urge you to look at it on how the genetic code was sort of set, right? How it came up. So we can discuss this a bit more, but yeah. So it's thought to have evolved under some degree of selection at least, but exactly how much and how much of a frozen accident it was at some point, not quite clear. Okay, so what you see here then is the relative use of leucine codons in different Vibrio genomes. Okay, these are different Vibrio cholerae genomes, or some from other species, Vibrio fischeri and so on, isolated from whatever different habitats. And you see very obviously and clearly that, for example, here, CTG is a codon that's used to encode leucine a lot, about half the time in these 4 genomes. But these other guys don't use CTG as much. Instead, they're using CTT a lot more. Yeah. So this is kind of what we mean by codon use bias. And it's a huge bias, you see, right? Like half the times you use leucine, which by the way is very commonly used in amino acids, you're ending up with CTG, and half the times you're ending up with CTT, which is a large difference. And so people have been obsessed with understanding why this is the case. And in general, early work or early thinking, yeah. Quite a lot actually as well. So one of the earliest observations also was that highly expressed and highly conserved genes tend to have stronger biases in codon use within the gene. So for example, ribosomal proteins, let's say CTT is seemingly favored in the genome of a particular bug. Ribosomal proteins will tend to have much more CTT, let's say 80%, 90% CTT of leucine amino acid codons, whereas other genes in the same genome may have 50, 60, 70. So again, this led to the idea or the expectation that it is selection which is favoring the use of specific codons, because highly expressed genes tend to be using more specific codons. Yeah? Whereas non-highly expressed genes, which presumably evolve under weaker selection in the genome, may end up using or seem to be not using these codons as frequently. They don't have as strong a bias. So they don't seem to care as much about which codon is being used. So that view was called the selectionist view, where the idea is selection favors or removes specific codons over time. The other alternative hypothesis, which was also very strongly favored, was a neutralist expectation, thinking that codon bias is something that emerges, and the processes that give rise to codon bias are not actually related to the fact of codon use. Okay, there's something else going on in the genome, and we'll come to some ideas of what may be going on, and that is what's going on. And so these are, of course, extreme camps, right? And as we discussed earlier, nothing is almost always true one direction or the other. There's a mix of both that are going on, and I want to highlight this in today's talk by going over Bulmer's model as an example. So Michael Bulmer made this very nice and very difficult, frustrating model. You'll see why, which tried to address as a function of mutation, selection, and drift, all three processes that could play a role in generating specific codon biases, to try and address the question of how much is selection actually acting on each particular codon and the codon use. Yeah? And I want to just go over some aspect of this model so you get an understanding of how this stuff and these kinds of questions connect with the kind of classical population genetics that you've learned so far. So the question then is, what is the value of S between codons? Okay, that's the selection coefficient. So if you assume an optimal codon for a given amino acid, let's imagine a 2-box amino acid, okay? So you have, let's say, B1 and B2 are your codons, which are basically your alleles, yeah? And their relative fitness are given by 1 and 1 minus S. So in this case, B2 is deleterious, okay? And the question is, how do we find out what is the value of S? Let's say there's a mutation rate that causes the movement between B1 and B2 and vice versa. So let's imagine in this direction it's U, in this direction it's V, yeah? And the frequency at time t0 of both of these codons is pt and qt in a given population. Everybody with me? This is stuff that you've done before. It's very similar to what are the models that we went over earlier, right? So now in one generation, you can calculate the allele frequency change, right? Due to selection, due to weak selection and mutation. So what he's assuming in this model is that selection is weak, right? Because it's obvious that if selection were not very weak or were very, very strong, you would see complete codon bias in everything. But you wouldn't see any variability, or you'd see very little variability. Because we do see quite a bit of variability, both in genomes and between genomes. Everybody okay? All right, so how do we calculate this? We calculate delta P, for example. And this is why I find this frustrating, is he has absolutely no explanation of where this came from, but I'm just going to give it to you. Okay, so in general, intuitively, I think most of you can understand where this is coming from, right? So this is the mutation rate going from B1 to B2, right? And so if you start off with frequency P, you're going to have that probability of generating B2, right? Similarly, the other way around, you have the opposite, the back mutation rate, which is V, that probability of losing B2 to become B1. And this is effectively saying, based on the value of S, how much am I going to alter the frequency of P in this population at this point of time, given the frequencies in the previous generation. Yeah? So then if we, and then, so this is all fine, but this is not really dealing with population size at all, which is ultimately what's going to determine what is the impact of drift versus selection. Right? So he brings in population size into this now, and he declares these two new variables. Big V is 2NE V, big U is 2NE U. You guys all right with intuition of why 2NE comes in? Why 2NE? We're considering a haploid case here. Yeah? So with the diploid, it would be 4NE. Okay. And then if big S is also 2NE small s, this is effectively a parameter that tells us what is the relative impact of selection versus drift, right? Given a finite population size. So the probability of fixation of a new mutant is calling this phi s, ultimately boils down to 2s over n, 1 minus e is to minus s. I'm not going to derive this because I have no idea how he derived this, so except that that's what he does in this paper. But I think it's interesting because it gives us some insights into what may be going on, right? And I want to go over some take-homes from this kind of relatively simple modeling that he's done. So in a large population, when u plus v is much bigger than 1, what does that mean if u and v are bigger than 1, much larger than 1? These are the values of u and v. What does that mean? What does that imply? High mutation rate, right? So basically there are many mutations that are going to be flipping back and forth between B1 and B2. We should see polymorphism at pretty much every codon position. Does that make sense to people? Yeah? So you won't see one codon fixed in the population when the mutation rate is very high. Reasonable, right? On the other hand, you will see a lot of polymorphism. But when the opposite is true, u plus v is much smaller than 1, it means mutations are relatively rare, right? And then you should see most sites are fixed. And when is this going to be the case? Under what conditions is the number of mutations that are being sampled very high? Either your mutation rates are high, u and v, or NE is very high, right? So in a very large population, again, you will end up sampling repeatedly all the different codons because of mutation. And so you will see more polymorphism at any codon site and across sites. Whereas in a small population, you are more likely to find fixed codons. And this is very parallel to what you've seen earlier with the other models that you went through, right? That effectively in a small population or when mutation rate is small, you're going to start seeing mutations behaving as if they are neutral, as if they are largely driven by drift. Whereas in large populations, selection takes over much more. So here he goes on to sort of calculate and make a more general model. So this is a more general model for what is the expected degree of codon bias in different populations. And then he goes on to develop a specific model in the same paper. I should write down the citation if somebody is interested. Then he goes on to develop a much more specific model where he asks now if selection is acting on codons because of their impact on translation rate or translation accuracy, both of which were dominant hypotheses at that point, what do we expect to see in terms of codon frequencies, right? So he uses all of this and ultimately predicts that selection on translation rate as well is going to end up reducing the degree of codon bias at the early parts of genes. Okay, this is a very nice prediction that he makes, a very specific prediction that comes out of the specific models that he makes, built upon these generalized models, and these are actually largely true. So when people had more genome sequences for a bunch of bacteria, people found that indeed the beginning parts of genes, the 5' ends of genes, do have less codon bias compared to the rest of the gene. And this is a very odd observation, right, and very hard to explain otherwise. And so your model is sort of helping you understand this or at least make sense of it to some extent. All right. So the general intuition is if translation rate is limiting, and basically what is translation rate? That you want to produce X amount of protein per unit time, right, for a growing cell to be able to grow properly. So selection is acting on the ability to produce a large amount of protein in a short time. You want to move the ribosome very quickly through the mRNA or the other way around. Yeah, so So effectively the idea is at the beginning, if you have a lot of stalling of ribosomes because of using rare codons, and because the rare codons map to rare tRNAs in the cell, then your ribosome stalling is going to reduce the rate a lot. And that is much more problematic at the beginning of the process. This is analogous to this traffic jam model that other people developed much more recently, and it shows effectively the same thing, right? That if you're going to have traffic jams on highways, it's much better to have the jams sort of interspersed throughout, rather than have one bottleneck where everybody's trying to get on the highway and everybody's trying to get off the highway at the same time. That jam is much more disastrous and will lead to ridiculous amounts of jams all the way through, instead of sort of spacing your entry and exit points throughout in highways. Vaguely, at least intuitively, it should make sense. Yeah, so they're basically, the speed is much slower at the beginning. So if things are a little bit slow but not jammed, then things are chugging along, and then you can speed up, and that leads to an overall faster speed. That's the general idea. And that's what his model suggests as well. Yeah. So if you're interested in these kinds of questions, do go back and read the paper and try and see what you can get out of it. Okay, all right. So that is just a glimpse of kinds of situations where population genetic models can help us predict or understand phenomena occurring at the level of genomes and how genomes evolve. And the big other general questions, oh sorry, I have to go over neutral theory first. Okay, so related concept to this idea of synonymous and non-synonymous mutations is Kimura's neutral theory, which was proposed in 1968. Just to give a little bit of background, how many of you actually have heard of Kimura's theory before? Everybody's heard. Okay, everybody knows about this, or should I fly through this? Okay, fine. I'll go at my own pace. If you're getting bored, you tell me, or if you're not understanding something, you tell me. Okay, so the general thinking when Kimura proposed this, so let me step back. People had been trying to understand how much variation exists in populations, right? So you've seen these pictures of, let's say, snails collected of one species at a particular place, a particular beach, and they're all over the place, right, in terms of their phenotype. So people clearly had known for centuries that there is a lot of phenotypic variation in populations. As some methods became available to understand the genotypic variation in populations using allozymes or small bits and pieces of DNA, fragments of DNA, which people could sequence around this time, 60s, early 60s, it also became obvious that there is quite a bit of genetic variation also present, right? And now if you remember, if the idea is there is selection acting on everything, all traits, all genotypes and so on, we should not see so much variation, right? So there was a lot of debate and sort of flux in this field of thinking about why are we seeing so much genetic variability in populations? Because selection should constantly be throwing out this variability, right? So people were trying to understand where does this variability come from, why is it maintained, what does it mean, and so on. And Kimura suggested that actually a lot of this variability that exists is neutral and it does not basically have any implications for adaptation right away, or it does not represent selection. Okay, and what is he saying effectively? That most mutations are effectively neutral and most polymorphism is unstable, in the sense there'll be new polymorphism that's constantly being generated because most of the alleles or most of the variants we see are acting effectively neutrally. And I like to make two distinctions here. One is that when the actual absolute measured fitness effect of a new mutation is, let's say, actually zero or close to zero, right? Where in whatever environment they're in, we would say that this allele is neutral, right? Is not visible to selection and should therefore behave as if it is under drift, correct? I mean, it will be under drift. So it will bounce around and sometimes it's lost, sometimes it's fixed in the population by chance, okay? The other situation is a bit more nuanced, and I think it's worth getting it very clearly in your heads, is that even under large populations or even under selection, alleles can behave as if they are effectively neutral. Why? Either because the value of small s is very, very small, right? So under weak selection, or the population size is very small. Yeah? So effectively, even a selection coefficient which is not negligible may lead a mutation or an allele to behave as if it is neutral if the population size is effectively small enough. Does that make sense? So two things determine what is going to be the fate of an allele. One is its value of S, right, which we know, and the second is NE. So if NE is small, even a larger value of S would not guarantee that this mutation acts as its S value suggests. Everybody with me? No, so Kavita discussed yesterday this idea of clonal interference or Hill-Robertson effects and so on, right? Where clearly there are dynamics that occur between alleles that are successively coming up, which are also beneficial, right? So that's a different category. But in general, if in the situation where mutation is very weak, as in mutation rates are not very high, you do expect, and selection coefficient is very strong, this allele will go to fixation or nearly fixation before the next mutation shows up. Right? So basically there are different regimes of values of each of these key parameters where you expect one or the other situation to take over and be much more important. Is everybody okay with what we just discussed? Yeah, you have a question? Somebody else had a question here? Okay, so there are effectively two reasons why an allele will behave and bounce around, right, as if it's in drift, if it's actually under drift, or if its population size is smaller. Yep. So what Kimura is suggesting then is that most of the mutations, the allele variation that we see in populations is effectively neutral, either because population sizes historically have been small for whatever species one is considering, or they really, their real S value in some sense is just very, very small. Okay, it's very close to zero. And this was hugely debated because at the moment the reigning idea, two reasons sort of, I'm condensing a lot of literature and history and drama here, but so many people were sort of very adaptationist or selectionist, okay, where they believe that most of what you see is driven by selection as an outcome of selection. And the second thing is, it was perceived that Kimura was suggesting there is no selection, okay, or selection plays a very negligible role, which is, again, huge amount of drama and debate, but that's not seemingly what he was suggesting, right? He was saying that the large fraction of mutations and polymorphism you see, or variation you see, is an outcome of these drift neutral dynamics. And of course, once in a while, adaptation and selection are going to take place and they're going to drive stuff to fixation and so on and so forth, okay? Okay, so then the other interesting outcome of this neutral theory is also the realization of the suggestion that if now, let's imagine population is tootling along, right? Nothing is happening. Okay, it's got a bunch of mutations. Okay, it keeps accumulating mutations. And then bam, the environment changes suddenly. Now this population has built this large sort of reservoir of accumulated, effectively neutral mutations, which serve as the standing genetic variation on which new selection pressures can act, right? So now when an environment changes, instead of waiting for that great mutation to come along, which is going to then, whatever, give rise to adaptation and get fixed in turn, you have already a pool of variants to work with, and maybe you're lucky and one of them is a jackpot, right? But was completely useless in the previous environment. You were lucky that it even was around still, right? So basically accumulated neutral mutations can then later become adaptive, or the other way around, they can be hugely deleterious also in a new environment, right? Yeah, okay, so it's up there. So is everybody okay with the core concept of neutral theory? Yeah. And so now this theory has lots of implications for how we do analyses of population sequences, okay, of genome sequences and so on. So now, at the moment, the way we see it, the way we work, at least in this field, is we say, okay, let's assume nothing is going on. Okay, let's assume that everything we see, all the variation is purely neutral. That's your null hypothesis. And now to claim that selection is acting, you must show something that's happening beyond what is expected by neutral theory. Yeah? So effectively, this then forms a very nice null model against which we can compare whatever we're seeing. Similar to what Hardy-Weinberg is, right? Hardy-Weinberg is like a null model in some sense, tells you nothing fancy is going on, nothing exciting in the population, this is what it should look like, this is what it should stay like, right? And then if something changes, then you're like, acha, kya ho raha hai? Very similar stuff. And the only some difference here is that we're working with genome sequences. So we're saying you walk along a genome, and let's say we sequence all of our genomes in this room, okay? And we have lots of money and great sequence quality, and we align all of our genomes, and we take a walk across our genomes. You'll find some sites where all of us are exactly identical, right? You'll find some sites where some of us are different, some of us are not different, okay, fine. Right? So basically, if this is sort of the root average going on, then where you see spikes, where you see lots of polymorphism, or where you see huge dips where there is no polymorphism, those then are become more interesting because they are then departures from this baseline neutral theory. Yeah, and I'm simplifying this. There's a lot more, but okay. Any questions about this so far? So sorry, in case people didn't hear the question is what about Ota's work? Tomoko Ota was Kimura's student, and she did a lot of work on this theory as well, to sort of help understand it better, and she continues to work on this stuff. So what Ota proposed was that actually it's not that most mutations are neutral, like absolute neutral, but they are nearly neutral. And her work suggests that she made a bunch of models that suggested that the dynamics of nearly neutral versus actually neutral mutations are interesting and different from each other, okay, in terms of driving adaptation or selection and so on and so forth. So we won't have time to go to the details of what exactly your theory is, but yeah, that's sort of the general summary of her work.
To coding regions of the genome, because the neutral regions will the non-coding regions may never have the opportunity to become adaptive.
Why?
Right?
What? Are you a codist?
No, what do the non-coding regions really do? Like
non-coding regions have regulatory elements, right?
But then they're still okay. They're still coding kind of in a way.
No, they're not coding.
Yeah, they're not coding, but
coding is protein coding. Okay, we'll come to that in a bit. Yeah. So how does this now relate to the synonymous, non-synonymous stuff? Obviously, the synonymous mutations, right, should be classic neutral ones because you don't change the amino acid. Their sort of S value should be perfectly zero. Yeah. And this is how we've functioned for a long time. We still continue to function like this. Where most of these tests, so you saw a lot of papers, right, where there were these log likelihood ratio tests and Many of them, you'll see DN by DS ratios being calculated. Effectively, DN DS ratios are saying, okay, given a gene sequence, if I account for the number of synonymous and non-synonymous substitutions that are possible, how many actual non-synonymous or synonymous mutations do I observe in my sample, right? And so you say, if the number of non-synonymous substitutions is very high, maybe this is under some kind of selection, this particular site, okay? So now all of this depends on this idea that synonymous mutations are neutral or behave effectively neutrally, either because the selection coefficients acting on each codon are very, very weak, very, very small, or because population sizes are too low. Yeah.
So taking the example of comparing the sequences that we do, right? So if a particular sequences you find it is conserved across everybody in this room, so how do you differentiate that is because of selection or because of our shared ancestry? So how do you delineate between the processes? Or
we need to have an outgroup to really make any conclusions for that. We'd have to have some chimpanzees in the mix.
Okay. Okay.
No, I mean, because
that is possible,
right?
Yeah. So, but that is possible.
Yeah, you're right. That you can't differentiate just based on what we see here. So you need to have some other comparison to ask what was the ancestral genotype like, right?
Okay. Somebody from far behind the ancestor. Okay.
I mean, there are many ways of getting at that, but that's one of them, for example. Yeah. Any other questions?
How do you explain selection on codons if the changes are synonymous?
Yeah, well, we don't have a very good answer for that. So let me come to that actually. I didn't come to that yet. So a large amount of work actually in the last decade or two has suggested that actually this is not true always, that synonymous mutations can face selection sometimes. And I've done some of this work where if I made a bunch of synonymous mutations in bacteria, it seemed to have a large fitness impact, right? They would grow or not grow or die or not die, whatever. So what seems to be the general upshot is that there are many different hypotheses that may explain in different contexts and in different organisms why specific synonymous codon changes are selected or not selected. One reason is this translation rate and accuracy kind of idea, which seems to be true in some cases. One possibility is the mRNA structure or stability. So when you change codons, you don't only change the amino acids or not change the amino acids, but you also change the mRNA sequence itself, right? The messenger RNA sequence, which twists and folds on itself and has a secondary structure. So that secondary structure is disturbed because you change the nucleotide sequence. Maybe that has a role to play, and there's again some suggestion that that is the case in some bacteria and some other organisms. Other situations suggest that there is selection acting on the nucleotide composition. Okay, so the genomic GC content, for example, of an organism, how many G's or C's it has versus A's or T's in the as a nucleotide base. Maybe that is under selection as a global property. And if that is under selection, in general, you will expect that GC-rich codons will be used more if GC is overall favored for some reason or the other, right? So there are many hypotheses, and it's not clear that there is one king hypothesis.
Easy to like check, right?
No, very difficult to check.
No, I mean, can't you look at codon usage and GC content across organisms?
Those are strongly correlated positively, as you'd expect. I mean, otherwise you won't get a high GC genome, right? In a high GC genome, you must use GC-rich codon, otherwise, right? So it's a bit tied together. But to show causally that selection on GC is what's driving this, very hard. Or is selection on codon usage driving GC changes? So much more difficult. So we've been trying to tease that apart. We've done some bioinformatic analyses in my lab to try and get at these questions. We don't have any answers. We have sort of partial answers. Yeah. Okay. So this is sort of generally where things stand. And more, there is very good evidence that synonymous mutations are not always neutral. But by and large, I still believe that that is the case, and that's a reasonable assumption to make. Continue with what you're doing at the moment. Yeah.
But you can, as in, a GC-rich genome is GC-rich across the whole thing, and the codon usage is only limited to a protein coding genes, right? So you can make a reasonable assumption that because the genome is GC-rich, that is why the tRNAs are more GC-oriented.
No, there are much more complications to that. One is in bacterial genomes, at least, the non-coding region is not very large. It's a very small fraction of the genome. So actually your genome GC is effectively your coding sequence GC. And there are other complications where tRNA genes and other components of the translation machinery seem to be evolving under their own selection pressures also, which are distinct, right? So this is why this is a complicated problem, and this is why it will keep me going and happy for a long time. Yeah. So we can talk more in detail. There are many interesting aspects of this. Fascinating. Why? The reason I said effectively neutral is because your classical simple definition of neutral would be S is 0, right? So if S is 0, sure, it will act neutrally, fine. But if NE is very small and S is not so small, so not 0, you may still have alleles behaving more or less neutrally. That's the point there that I wanted to stress.
If there's a mutation which probably currently doesn't affect an individual but might have some later life implication, do we call them neutral mutation, or what do we exactly call them?
Point. So Basavi is asking about mutations which may be beneficial or deleterious early in life, but have a different impact later in life and so on, right? For the same organism. And we think there are some such mutations that are known, right? This is one major idea why, one major hypothesis for explaining why aging happens, is that mutations that are beneficial for us early in life are fixed in the population or are favored, but then those same mutations have a deleterious effect later in life, and that's maybe effectively why we all get old and die. Yeah. So what do you call these mutations? You know, difficult question. I don't know. I mean, it's clear that it's not a simple, yeah. So I guess you would have to define it based on what you are looking at. So if you're asking about reproduction or survival, then this mutation is either beneficial or deleterious, as the case may be. If you're looking at lifespan, then yeah, what is proper reproduction? You're getting yourself into a larger and larger hole. Okay, how about we discuss this a little bit later? I want to get through this. If we have time, we'll come back to this. Okay. All right. So, what are the sort of some of the major questions in genome evolution that people are trying to address? And some of these are open, some of these are whatever. There's a lot of nuance here. So in general, what aspects of genome architecture are shaped by natural selection and under what conditions? When do we expect that genome GC content will be under selection? Is it fair to say it's a trait that is under selection and so on? Lots of issues. There's a lot of work trying to understand the evolution of regulatory elements, which is the sequences which are non-coding bits of DNA upstream of a coding sequence. And these are thought to be, for example, regulatory elements, promoters, places where ribosomes will bind and so on and so forth, right? And so how much of evolutionary changes are driven by changes in regulatory elements like mutations in this non-coding area versus mutations in the coding sequences? This is a question that people have been obsessed with for quite some time as well. I think it's sort of dying down slowly because there are clear examples of all kinds of these, right? As we get more and more data. But this is one question that has motivated sort of population geneticists for a long time to understand what kind of mutations, where in the genome do you get the biggest bang for your buck. Okay, another set of questions has focused on understanding gene copy number and position. So again, effectively all of this is in some sense a little bit like natural history work, right? Like you saw the earliest natural historians sort of go out and see all this variation and start wondering, why do I see this variation? What does it mean, right? Is it good for the species, bad for the species, good for the individual, bad for the individual, all of that. Similar ideas here with genomes, right? So we see a huge diversity of genomes. We see a diversity of gene copy numbers. There are some genes that are in hundreds of copies in some genomes, and some genes that only have one copy but are super essential, right? So does that matter? Is that something that's shaped by selection? Is the position of genes something that is shaped by selection in a genome? If I have a very, very essential gene, should it be on chromosome 1? Should it be on chromosome 10? Does it matter at all? Who should it be next to, right? And so on. These are the questions that people are asking as well. People are asking questions about nucleotide composition, like GC content. Is this under selection? If so, what is the selection strength acting on it? What does it depend on? How does it vary across prokaryotes, eukaryotes, and so on? And finally, mobile genetic elements, which are a really special and very interesting class of bit of DNA, right, which can move, and it can often move itself. So it codes the machinery to be able to cleave itself and then duplicate it or insert itself into another place in the genome. So again, there's been debate about, are these selfish genetic elements, right? Because they basically can replicate more than what the rest of the average genome replicates, the rate at which it replicates. Are they parasitic or are they beneficial? And the genomes keep them around because there's a benefit to be had from these mobile elements sometimes, right? Because if you think about it, mobile elements also give rise to genetic variability, right? Because they would sort of move around in the genome and create havoc sometimes, but also create perhaps some new interesting combinations of genes next to each other or not. Yeah. So just a broad overview of sets of questions, sets of approaches which people can use to understand how genomes themselves are evolving, not just as sort of agents of evolution, but as selected traits potentially in of themselves. And of course, the baseline here always is saying how much of this is selection and how much is sort of neutral genetic drift. Okay, so I want to quickly move to a little bit of stuff about Evo-Devo. And this is sort of evolutionary developmental biology, and you'll see why I'm interested in this and talking about this a bit. So you see a lot of major phenotypic changes evolving during development across the animal or plant phylogeny, right? So for example, body plans and segmentation. Is everybody familiar with what this means? Yeah, a basic body plan of sort of how many segments you will have. Each of us are segmented, believe it or not. You have segments, you have a vertebrae, right? And they all develop in a very parallel fashion across the animal world. How do such changes occur? You know, it seems like such a nice, beautiful, regulated machinery. How do all these parts come together? How does it evolve? If you talk to any proponents of sort of non-evolutionary change, like people who don't believe that evolution explains the variation we see around us, this is a favorite example, right? This kind of stuff, like, oh my God, this is so intricate. How is it possible that effectively random chance has given rise to this kind of intricacy and complexity? So the short answer is there are sets of genes that that very commonly are involved in these sort of large changes that occur in the body plan or in development. And these changes, as an example, are driven by these sets of genes called Hox genes. homeobox genes, which are a class of homeotic selector genes. Don't worry about the terminology. Basically, these are sets of genes that control patterning of body structures, right? So segment identity, that my head should be on one end and the tail end should be the other end, and what comes in between and which segment comes after which other segment. Where should my limb develop, right? My limb should not develop from my middle segment. It should develop from this sort of more anterior segment and so on. So those kinds of things in metazoans are regulated by homeobox genes, and they share, all of them share a motif that binds DNA, and this is why they become so critical and important for major evolutionary changes, right? Because all of these sets of genes, there's a bunch of genes that are paralogs. Are we familiar with paralogs? Paralogs are basically copies of a gene that are present in the genome. That duplication and that copying event happened much earlier, and now these different copies of the same gene have started to look a little bit different and may have taken on different functions. Okay, so the class of homeobox genes, Hox genes, are such paralogs. I'll show you some data which sort of makes this clear. But effectively, all of them have a little piece in the protein which binds DNA, and that is how a single Hox gene can regulate the expression of lots of other genes, which makes them really important sort of master puppeteers. Yeah. Okay, so here's an example of what happens if you mess with Hox genes. These are these famous experiments where there's a fly head, and that's the normal fly with a normal antenna. And if you disrupt some of the Hox genes which are responsible for the production of this antenna here, you might end up with a leg in place of an antenna, right? So it's very, very bizarre stuff that happens. So what that says is, look, the machinery to produce a leg effectively exists in all of these places, right? But what makes it an antenna and what makes it a leg is that one little key that has to be a little bit differently tweaked, and you get one or the other. That makes sense? So that basic body plan is sort of there, it's preserved. You also end up getting things like this, where flies are dipterans, they have only 2 wings, but other insects, the sort of ancestors of these groups have 4 wings. So these guys also have these vestigial little body things called halteres. And if you again mess with Hox genes, you will end up getting a 4-winged fly, right? So where the halteres, instead of being these reduced sort of wing structures, will develop into full-fledged wings. Again, the plan is there, you just need to tweak a few things to get it to work. And so this is just a micro, I don't know if this is a true image or a fake colored one, but whatever. The point is, I mean, it's definitely colored fake, but the image whether it's true. Anyway, so this is kind of an egg of a Drosophila, right, of a fly. And they've painted it to show you where each of these different Hox genes or genes in that regulatory pathway are being expressed. So you see these rings of, or these lines of expression, right? And if you again swap these around, you end up messing with how the animal is going to develop. There are examples of what happens in digit development. I think these are mice. So yeah, so here's your normal digits, and then you get the weird mutations showing up, again with very small mutations in Hox genes, or how they're expressed or where they're expressed, right? So these are very, very important genes, and this is kind of a blueprint for saying, well, to get these large phenotypic changes, right, going from this very strange digit morphology to your normal stuff, you don't need to do much. You just need a few mutations in these very important genes and you're set. Yeah? So evolution can jump through such kinds of changes in such kinds of genes. So this is a phylogeny of animals showing you Hox genes. So the order of Hox genes is conserved. So this is, if you look across the genome, which one comes first and which one comes later. The colors represent different paralogs of a Hox gene, right? So the red is thought to be the most ancient copy of this one particular Hox gene that's present in all of these different animals. Now, within here, let's say the orange guy duplicated from this guy duplicated from this guy. That's not necessarily true, but basically there have been a series of duplication events that occurred. But all of these guys are homologs to each other, right? So those duplication events happened in the common ancestor of all of these animals. So you have these basic set of Hox genes which were present in the common ancestor of all of these animals. And then since then they've diverged a little bit. So are you okay with this? I see some blank faces. Yeah? And the body plan itself is also largely conserved. So where the red guy is going to get expressed is towards the anterior end in all of these different animals, right? And the purple one is always going to be expressed towards the tail end, and that's also more or less conserved across the animals. In some cases, you see these other interesting cases where there have been sort of further duplication events within this lineage alone, right? So now there are 4 copies of the red gene, of the red type of the Hox gene in this guy, and tetrapods also similarly have 3 copies of this and so on. Yeah? So this is effectively saying how relatively simple but major events can change a lot of important phenotypes in animals. Okay, so in general, there are conserved developmental pathways which can allow these kinds of changes. There are global transcription factors. Transcription factors are proteins which will allow or regulate the expression of a particular gene at a particular time or in response to particular cues and so on. So again, transcription factors can be some of these major keys, right, through which you can turn and easily mutate to cause a lot of changes in expression of lots of different genes. And they can control organ development, like distal less is one such gene, I less is one such gene. These are genes that are very well studied and understood in some insects or other model organisms. And regulation of different genes at different times controls developmental variation. I wrote down enhancers here, but let's not get into that. Forget about this for now. And both cis and trans regulation play a role. The regulation may be, let's forget about this for now as well, actually. And development is modular, which is another thing or another way in which major changes can happen, right? So this sort of segmentation is sort of modular. There's one module, you go through it, there's a protocol, there's a program, you follow it, right? And so you have to tweak the instructions a little bit. If your basic script is a little bit tweaked, you get this whole module behaving differently, right? But then that's independent of other modules. And so the other modules can also be tweaked independently. And together, if you tweak all of them, you'll end up getting much more variation. This is a bit abstract, I understand, but I just wanted to give you a sense for how these big changes can occur. Here's an example where genes are also co-opted for new functions. And this is a very, turning out to be a common theme in evolutionary biology as well. This is an example from butterfly eye spots. So I'm sorry you can't see the variation very well, but this is a dry season form. This is a wet season form. So it basically allows it to have better camouflage in different seasons when the surrounding vegetation is dry or wet. And when you zoom in into these eye spots, these structures, you see the pattern is kind of there, but the elements of the pattern are changed, right? So the white spot is bigger here compared to this one. There's a much darker spot here. There's not much dark spot here. So basically the dark spots are all melanin, which is a pigment which is deposited or not. And what they basically show is if you overexpress this distal less gene in the larva, the developing caterpillar, you end up getting these different patterns, right? So just by changing the expression of one gene, which by the way does other jobs, it's normally involved in neural development and so on, it's known to be involved, it does these other jobs as well, right? So because this gene is everywhere in this organism, is used a lot, now you can tweak just when and where exactly it's being expressed, and it then controls a bunch of other things, which lead to all these phenotypes which have nothing to do with neural development. Make sense? So this is what's called exaptation, where you have a new use for an old trait or old morphology. For example, bird feathers were used for thermoregulation first, we think, and then flight, their use in flight evolved later. Yeah? So feathers are good insulation material. So we think that's what evolved first. Right? So this is called exaptation, where classically, and the boundaries here and the semantics are a bit murky, but in general, exaptation effectively means that this trait may or may not have some use right now, but it's definitely getting a use a bit later, right? So you sort of pre-adapted in a vague sense. Everybody fine with this? Okay, so then just a couple more examples of sort of novelty that has evolved. These butterflies have uric acid on their scales, which makes them this white color, and that's an exaptation, right? So uric acid was being produced, is produced in all these insects. They don't have sort of mechanisms to get rid of the uric acid except in their pee or poop. And basically uric acid is now sequestered and put on scales to give it this white appearance, helps in camouflage, helps in evading predators and so on. In this salamander, the loss of lungs removed a constraint on tongue, right? So look at how long its tongue is, and the tongue has to fit in its mouth somewhere, right? So actually it's partly near to where its lungs would have been. But now that it lost its lungs when it became a more amphibian, it had space for this longer tongue, and that longer tongue can fit there. A bee sting, has anybody been stung by a bee? Is actually a modified ovipositor, which is the appendages that it's used to lay eggs, right? So basically stings and egg-laying apparatus has nothing to do with each other, right, you'd think. But they're actually modified forms of each other. So it's very, very interesting. So such new traits and novelty in sort of what you see can evolve by slight modification of existing stuff because things are modular, because developmental pathways are conserved, because there are these global regulators that can be changed relatively easily instead of changing the whole machinery. Yeah? Are you with me? Okay. So in general, all of this sounds great, but then there are also constraints on evolution, right? You may have physical constraints. For example, oxygen diffusion, we think limits insect size at this point because they don't have sort of a circulatory system that, like ours, they don't have lungs and so on. So they're basically dependent on oxygen diffusing in through their spiracles and making its way through the body. So if your body is too large, you don't have enough oxygen making its way around. So your internal organs don't get enough oxygen. You may have selective or functional constraints. For example, pleiotropic genes, where one gene has multiple effects on different phenotypes, right? So if there's selection on one phenotype, then the evolution of this gene with respect to its impact on another phenotype may be constrained. Does that make sense? Yeah. So pleiotropic genes may face such constraint. There may also be genetic constraints. There may be a lack of variation for butterfly pigmentation gene across different body parts. So there are some body parts of butterflies which do show lots of pigmentation variability, right? There are like wings, they show lots of variation in color pattern. Why doesn't the body show so much color pattern? Why don't some butterflies don't show any variation at all? And so it's attributed to some degree, in some cases specifically, to lack of genetic variation. So if you don't have variation for producing different color pigments in all parts of your body, you cannot do it everywhere in your body, right? So you may not have variability that's relevant always. And you may also have developmental constraints. So for example, is everybody familiar with stem cells? What are stem cells? Huh? What are stem cells? Do you know what are stem cells? Anybody who does not know what are stem cells? Really? Okay, good. So then I won't talk about this. Does it make sense? So stem cells are lineages of cells that are able to differentiate, right? So most of us don't have many stem cells left in our bodies anymore. We've finished our sort of differentiation and growth period. We have very few stem cells in very specific positions in our bodies. But babies, fetuses, they all have lots of stem cells because they're still differentiating, forming new tissues and organs. But, and so it's thought that this lack of stem cells in an adult is effectively what limits evolution of some traits at that age, right? So if you're able to modify stem cells at a particular age, you can have the development of even new and more phenotypic variability, which is limiting for adult polyforms. Okay, I think that's it. Yeah, no, so it's just that, so it basically means that then the stem cells have to differentiate and do their job earlier in life when they're actually not under selection, right? So the selection is acting on the phenotype at a later point in life. And so you basically need much more programming to have happened to make sure that the setup is there, but the development only happens later, right? So because tissue differentiation has to, let's say it's favored to have an ear here or something, right? But to develop an ear here as an adult, you need to, I mean, you can't form ear cells. I don't know enough about stem cells at this point, but the point is that you can't form that now at this point in your life because you don't have the right kind of cells. And when the cells are present, it's not favored.
But if, for instance, it doesn't matter at that point of time,
then it's fine.
Even if
yeah.
it will happen.
Yeah.
Only if there are two different
yeah.
selection pressures at those 2 points.
Where it's easiest to imagine how this would be a constraint, yeah. Okay, I finished on time. Any questions? Okay, so this lecture was meant to be sort of a broad overview of where do population genetics and molecular evolution converge? What are the new questions that are in front of you, open for any of you who want to do more work? And in the next slot, I forgot to mention earlier, I switched the afternoon slots with Uma. So Uma will do the before lunch and after lunch session, and then I'll finish up with an exercise at the very last one today. Yep. I'm sure you don't care, but okay, all right. So we can wander and get some coffee in 5 minutes.